Mr. Shah, sir? Hello.
Yes, sir. Yes, sir.
So students join ಆಗ್ತಾ ಇದ್ದಾರೆ ಸರ್ ಒಬ್ಬೊಬ್ಬರಾಗಿ. ಹಲೋ. ಹಲೋ ಸರ್. ಶುರು ಮಾಡೋದ ಸರ್? ಹಲೋ. ಹಲೋ.
ಹಾ ಸರ್.
ಶುರು ಮಾಡೋದ ಸರ್?
ಮಾಡಿ ಸರ್, ಮಾಡಿ.
So
If students are there, I have no issues. I am ready, sir.
Join ಆಗ್ತಾ ಇದ್ದಾರೆ. So I will give some small introduction about you, sir.
Yes, sir.
So I welcome to today's talk resource expert Santana sir. So good evening everybody. So and I also welcome to our Chief Executive Officer Dr. Ramesh sir and our academic president Professor Ayappan sir. So I will give a small introduction about Santana sir, Program Coordinator of ACTS at 75 Students Satellite Mission of India, Secretary of NBSF, North Bangalore Science Forum, First Column Library in South India, and Head Department of Biotechnology at SFGC. Is a biotechnologist by profession and environmentalist by passion. So, nurturing and motivating young talented minds towards creating a livable planet. He is very passionate for creating society with compassion, empathy, and humanity. He is presently working in as management for the Green Karnataka initiative for creating smart and clean cities. A master trainer for waste segregation, exposure, and management for smart cities. He is looking forward in creating a behind of young, talented, and ignited minds with ultra-modern techniques and traditional Indian value system for building nation with YPLBCT for sustainable development in conservation and preservation of Mother Earth. Thank you, over to sir. Hello.
Okay, sir. Yes, sir.
Thank you, over to you, sir.
Okay. So I wholeheartedly thank Karnataka Science and Technology Academy and exclusively my very, very close friend, CEO, Dr. Ramesh sir. Then our chairman for KST, Ayappa sir, and the program coordinator, Vidwat Gaura sir, who has given me this wonderful opportunity to interact with the students for the Science Talent Search program, which is a webinar under Government of Karnataka and KSTEPS and KSTA. The topic what I had chosen, the human health and disease, it takes me back to 2001 and prior to during my MSc days, where it was my dream. I was thinking that let me work on HIV. And I was lucky that I could get opportunity to work under ICMR director, the then Srikant Tripathi sir. And that's how I got interested in human health and disease. And this COVID pandemic last 2 years has made us aware of how important human health is there because of this frequent infestation of virus, bacteria, and other organisms, how we are very much exposed to. To first take care of this human health, what we must learn that, my dear students, we have to take care of this immune system first. So let me share the slide, then it becomes easy for me to explain. So hope is it clear now? Yes. So
Sir,
let
visible,
the
visible.
video. So here I could share with you and tell that immunology.
Yes, sir, immunology.
It's visible, sir?
Yes, sir, visible.
Okay.
Visible.
So here, what I mean to say that the word immunology means immune and logy. We can break down it into two different parts. One is immune, that is immunity or immuneness we call, and the logy is study. And it is a broad field. It's not like a very simple field. It's involved so many things. And it involves both the basic as well as the clinical applications. Take an example. If somebody speaks something to us, it depends on how we react. We may react rudely, we may react politely, or we may become silent. Same way in our body, when a mosquito bites, there will be different kinds of reactions. Many person you will see that they will just kill the mosquito, or just they will one blow of air and the mosquito will go away. Some people, they will keep scratching their hand, or some people will start saying, oh no, it has swollen the redness. So these different reactions, what does it indicate? It indicates that we have an immune system, especially with the vertebrates. We call it as a vertebrates. That means we have a spinal cord, we have a blood vessels, we have the systems, we have WBCs, red blood cells, defense systems. Like a country with, we can always compare immunity. I always tell my students, always compare immunity to as a war front. So every time we are in war. Now what happens, antigens, with the basic category, what we should understand is, one is antigen, one is antibody. Under immune system, two things are there. Like how we tell for reaction, one product and one enzyme and one substrate. So here same way, there must be, to give a sound, we require two hands to be. Like when we do like this, that means sound comes. But when we do like this, sound doesn't come. So here antigens and antibodies should react, then only we can see the reactions. So the first of all, antigens. What are antigens? The particles which are foreign, anti, against, foreign. They are not recognized by our WBCs during our generation. That means our formation in the mother's womb to birth and life. That means when inside mother's womb there is an egg and the sperm goes and fertilized, from that time, from a 2N cell, a single cell, zygotic cell, we glastula, then so many metamorphoses, the tissues, organ system, and so many things are happening. So during this time, our body has come across different cells in the body, and they are known as self. The moment our body come across any foreign particle, we recognize as an antigen, provided that the particle is capable of causing disease, which can be parasite, virus, bacteria, pollens. So like mushrooms, somebody they are allergic, eggs they are allergic, they are allergic to brinjals, they are allergic to the pollen, they are allergic to the chalk dust. So like this, these are the foreign particles. This is called as a classical mediated pathway, that is called humoral mediated pathway. Another one is called as cellular immunity, where we could see that our own cell become foreign. Like we sometimes tell, my friend has become foe. So never make a friend an enemy, because they will know all your loopholes. So same way, our own cell, like in cancerous cell, our own cell becomes dangerous. Arthritis, rheumatoid arthritis, our own cell become altered cell, we call that in scientific terms. So they know all our, so that time the antibodies doesn't work at all. That time they require cellular immunity. So these are the basic difference between humoral immunity and cellular immunity. So humoral means where antigen enters, antibody is released, and we get cured. But when it comes cellular immunity, that means humoral immunity is not capable of doing. Our cell has become altered cell and they are causing the problem. So this we must understand exogenous and endogenous. So repeatedly you might have seen or heard that terrorists are of two types. One is external, one is internal, right? So that's what I always compare India and body, our country and our own body system. So one, we fight, continuously fighting from external part, external microbes, exogenous, and some we compare or we keep fighting with endogenous, that is tumorous cells. Then if we go ahead with innate immunity, innate means these are born along from the mother, we inherit that, innate. Natural, they are non-adaptive. So therefore you might have seen the moment we are born, at least for 6 months, we are immune the moment we drink the first milk of mother, that is called colostrum, which has got highly rich in immunoglobulin A. Then comes the adaptive immunity, acquired, that is called adapt. We adapt according to the situation. That's why it is called as acquired or specific immunity. So overall here we will understand what is immunity to disease, hypersensitivity to biological reactions, allergies, and reactions of foreign tissues. So some more definitions if we go. What are antigens? So antigens are usually proteins from a pathogen or foreign organism that provoke specific immune response. See, we are very silent, quiet. Sometimes some people speak something, that's what I told in the, either we may overreact to it, we may fight, we may speak, we may be silent, or we may just smile. So same way, a particle, a molecule which is protein in nature. So you must understand an antigen. What is an antigen? It is a protein in nature, minimum 10,000 kilodalton weight, and it has a property of immunogenicity and immunoreactivity. What is immunogenicity and immunoreactivity? One is capable of triggering the reaction. Take an example of a teacher. A teacher motivates to learn. Student gets motivated. Immunogenicity. Immunoreactivity. Now, once the student gets motivated, he or she must continue learning process. If not, then we call there is no reactivity. So therefore, antigen is also like that. A antigen must trigger the genicity. It must have the capacity of triggering and completing the reaction of producing till antibodies or triggering the cell-mediated immunity. So therefore, it must have the specific immune response. Then what are antibodies? They are immunoglobulins. They are immunoglobulins in nature. And what they do? They are glycoproteins molecules. They are glycoproteins. What does glycoprotein means? Along with protein, glucose or lipoproteins they may be, or they may be still metalloproteins, anything, especially the immunoglobulins. Why we call glycoproteins? Majority of the immunoglobulins are glycoproteins. The reason is they have a carbohydrate moiety in the constant region or the stem region. And they are produced by plasma cells. And what they are produced by? They are produced by plasma cells. So what is a plasma cells? So when the B cell multiplies, they will be, right, when the antibody enters, briefly I'll make you understand. When the antigen enters into the body, then immediately our body has got macrophages, neutrophils. What they do, they go and capture the antigen. Then they decide what to be done. They engulf them by macrophasic action. And once they macrophage, what they do, they degrade them, phagosome, phagolysosome, and then they degrade, and then through help of MSC2 molecules, they project it to T cells. T cells then in turn get divided, activated and divided into T helper cell and TC cells, cytotoxic T cells. Now these T helper cells, what they do, they are like masters, or ring masters. What they do, then they now look out for, okay, this time what happens, like how these processes happen, the B cell again goes and binds with the same antigen and comes back, and T cell now interacts with the B cell and proliferates the B cell into memory cell and plasma cells. The plasma cells are capable of producing secretory antibodies, and the memory cells are retained for the future process. So that second time, if there is any attack, immediately there will be an immediate response. Then what is this response? That's what the third line of defense involves the production of antibodies and generation of specialized lymphocyte against the specific antigens. See, you might have seen the moment a person, right, gets affected with chickenpox, smallpox, diphtheria, all those things, they again never get back those, because they're in their body already the antibodies are there, because of which they are resistant to. Therefore, we call it as a specialized lymphocyte memory cells. So this is how, in a brief, you can see the first line of defense. What is that? Skin, mucous membrane. So you could see any reason, either from our mouth, nose till the anal part or anus aperture, it is lined with mucus. And you might have seen certain advertisements of cough syrup, right? They do, and they will show that no, the viruses like demons are there. The mucous lining are the one which protects us, right? From, because this nose we breathe, mouth we eat, whatever, or drink, many microorganism goes through. But directly the inner epithelial lining of the gut linings doesn't get exposed because of those mucus. So mucus protects us. Then eye, you see, immediately anything enters into the eye, tear comes out. So it protects us. Skin, you see, if there is a cut or wound, you could see there will be infection. Then the second line of defense is phagocytic white blood cells. Why we call phagocytic? Because they have phagosome in them. They are macrophasic in nature, and they have antimicrobial proteins, and then the inflammatory response. So what is this inflammatory response? What is this inflammatory response? The inflammatory response means if a mosquito bites, there will be four things: rigor, redness; calor, temperature; rigor, calor; dolor, dolor means pain; and temperature. And then the temperature. And the temperature. So rigor, calor, dolor, tumor. Rigor is redness, calor is temperature, tumor is swelling, and dolor is the pain. So these four things happens for any reason. You could see that either a thorn pricks, a mosquito bites, or there is a cut or wound. And the same thing happens when this, why the inflammation happens? During the inflammation, therefore the inflammatory response, right? If this inflammatory response is there, see, if someone punches us, immediately there is swelling. That time what happens? The internal damages get reduced. But if there is no swelling, that means the internal damages happens. Then the third line of defense is lymphocyte and antibodies, which is more specific to, with. So briefly, to go ahead with a little more non-specific barriers are, as I have told you, skin, mucous membrane. Then you have the physiological barriers. What these physiological barriers are? Lysozyme, lactoferrin, low pH in stomach, right? We see that we take lots of prevention of stasis, peristalsis. So what we could see that we eat sambar, we eat rasam. That means why I am telling those comparison. There is two different kind of pH. We eat sweet, we eat khara, we eat spicy, we eat chili. But what is happening? We are safe enough, our body withstand that. as it withstands, what happens, our stasis is maintained. Right? And the next part is our stomach, you could see as it is having low pH. So because of the lysozyme and lactoferrin, so many organisms, microorganisms which are not so much a pathogenic, they are not able to grow in our body. Then clinical relevance, you could see urinary infection with urinary obstruction, then bronchitis, culinary, and then we cough, right? That's what I was been telling, coughing. Because of the mucus, there is an irritation. We try to expel out. And if we cough, we see that the mucus comes out and it brings along the microorganism. These are all adaptive. Then comes the non-adaptive. What does non-adaptive means? They are called retiloendothelial system, monocular phagocytic cells. They are in blood. They are lymphoid, liver, spleen, bone marrow, lungs. And they are efficient in uptake and removal of particulate matter. They are non-adaptive. They are bound to, they are like clearing agents. Anything comes like security guards, like NSG commandos, black cats. Immediately anything of unknown comes, they will take away. Now one question comes here, how we can tell them as an unknown, right? How we know? Take an example, one army officer gets trained, gets selected and goes for training in a, suppose taken NDA, National Defense Academy. From here, while he going, he comes, he knows all his family, friends, relative, classmates, everything. Once he goes there in National Defense Academy, gets trained with, comes back again, does he forget the friends, family? No. But he knows then who are our real friends, who are our family, who are our citizens, who are our enemies. Same way in our body, from the bone marrow, this WBC or that is B cell and T cell, they gets matured either in thymus, that's why they are called as T cell, and they go to colloquial region that is called bursa fabricus in pigeon, and in our body lymph node, they become B cell. So while they come back, they know which all are our self cell, which are our non-self cell. So they try to remove the non-selves. They try to remove the non-self cells. Then comes the alternative pathway of complement. Because of this complement, we are safe to parasites, endotoxins, and what they do, they exactly do the drilling effect. So this pathway, it's in fact a long procedure complement system, like how we have seen with blood clotting factors. Same way we have the different complement factors where C3, C4, C5, 2 are there, 1 is there, then joins with 5, 6, 7, and 8, and 9, then they form a drilling complexes and drills the bacteria or makes, in fact, drilling in the sense, scientifically I can tell, they make pore in the cell membrane of the organism and they die because of the lysis. Then comes the phagocytosis, this engulfment that is called cell eating. Pinocytosis, cell drinking, engulfing and killing of the microbes by phagocytic cells, which include macrophage. And these microphage and macrophage. So these macrophages are there in different forms. Microglial cells, Kupffer cells, right? Different organs has got their own kind of clearing agents like bouncers. The process of killing includes both oxidative and non-oxidative mechanisms. Then comes the inflammatory response, already we have told, and fevers. So usually people are scared, okay, no, fever is coming, it's a problem. But we must understand, if fever is coming means, that means your body is responding. Endotoxins and interleukins, there are so many interleukins. There are so many CD4 and CD8. Even a patient is affected with HIV, the person won't die until and unless CD4 counts goes low below 200. Then comes the interferon. What is interferon? Virus-induced protein that have antiviral activity after 48 hours of viral infection. So therefore, during COVID, what they told, or they have told that after 2 days only, third day you should go for testing. 48 hours of viral infection. And then that is interferon alpha, interferon beta, and interferon gamma. And then the natural killer cells. There lies the tumor cells and virus infected cells. So this prevention of entry of organism, how we do? This is very simple process. Our body or our cell produces an opsonin. What is an opsonin? A plasma protein binding bacteria, like we catch or trap rats, right? What we do, we pick up a poison, put it into bonda bhajis or right, some breads or something like that. And then the rat gets attracted to it. And then we put either a jelly or right, what is called a gel trap, or we put that bones we call, or the trapping instruments or the metallic boxes. So opsonin is the one which helps in getting the organism adhere, because the cells doesn't have hands and legs and eyes, but they have senses, surfaces. So they for adhesion. So it promotes the adhesion of the bacteria, right, with the cells. And once then comes the process called chemotaxis, right, by which the degradation and other parts starts. Chemically attracted, attached, and then they are engulfed, and then they are being broken down into. So complement and phagocyte exist mainly in the blood. So a mechanism is required that is called inflammatory response. So any organism which enters into our body, they will undergo this process. So briefly to tell you what is innate or genetic, immunity and the organism is born with. They are genetically determined. So you see, if mother is too strong, immunity, usually the best example I'll tell you about the adaptive after some slide. So later, if you could see, therefore they tell when a baby is in mother's womb, mother is always told to take care of her health, because if mother is weak, even the child will become weak. This is because genetically determined. Maybe due to lack of receptor or other molecules required for infection, or innate human immunity to the canine distemper, or immunity of mice to, see, mice is resistant to poliovirus. And then comes this acquired immunity. You might have seen, right? Especially the workers on the road or with the cement. You see their childs are on the road, they are playing with cement, but touch wood, by grace of God, they are very safe with. Whereas the moment we so-called, modern, ultra-modern, and we stay within the four walls, the moment we are little bit exposed to cold or little bit exposed to the cement, we get affected with. That means during our day-to-day life, day-to-day work, we are not acquiring the immunity. So therefore, they tell little bit exposed. See, we drench in rain, we get cold. But do you think of the swimmers who are frequently throughout the day, 14 hours, 12 hours, they are in water, but they don't catch cold. So this is non-genetically determined, it is acquired. That's what I mean to say, they are acquired. And maybe acquired naturally or artificially. Either naturally it is acquired or artificially it gets acquired with. Then development of immunity to measles in response to infection or vaccination, measles, mumps, and this is the one which you could see. These are the different parts of the body where we have like lichens, lymph nodes, tonsils, right, spleen, Peyer's patch, thoracic duct, appendix, lymphatic vesicles. You could see these are the parts where we have lymph nodes. And these lymph nodes are the one where our B cell gets matured with. So what is the type of acquired immunity? So naturally acquired active immunity, naturally acquired. So first we'll go with acquired immunity. Enter body naturally, antigens, like we getting exposed to. Body generates an immune response to the antigens. And it's a lifelong, like chicken pox or mumps, or if it is a temporary influenza, influenza is always temporary, or intestinal infection. And natural acquired immunity. Why it is called acquired immunity? Naturally acquired passive immunity. That means here we are not capable of directly producing the antibodies, but we are taking the antibodies. Antibodies are getting passed. Therefore, we call it as a passive immunity. Active immunity means like you could have seen in COVID, plasma therapy. That means the person, why plasma? Plasma has got antibodies. So when a person is not capable of producing antibodies, their antibodies has to be entered. Even in dengue, you could see that, right? Or platelets are been. That means the person is not capable of producing platelets, generating high amount of platelets. So it has to be supplemented. So antibodies pass from mother to fetus by placenta or breastfeeding. That is called colostrum. No immune response to antigen. There won't be any response, because if there is a response, B cell, T cell involvement should be there, but there is no. Immunity is usually very short-lived because you are borrowing from somebody else. So it is short-lived. It can't be lifelong. And protection until child's immune system develops. So therefore, after 6 months, the child, the moment he or she gets exposed to the first food, that time the child slowly starts building their own immunity. That is called acquired immunity. Then types of acquired immunity: artificially acquired. How you can artificially acquire? Receive a vaccine or immune serum. Receive a vaccine. Artificial active. What you do? Antigens are introduced. Like, take an example, what we do. Now during COVID, what is happening? They are injecting a part or fragment of antigen into our body. Or in polio, a fragment of the polio virus is entered, and therefore we start generating antibodies. Therefore, it is called as an active immunity, artificially acquired. It's not naturally, it artificially been injected into our body. Then body generates the immune response to the antigen. And then immunity can be lifelong, like polio vaccine, or temporary for 2 to 3 years, like tetanus toxoid. So every 3 years, we have to keep taking the tetanus. And then artificially acquired passive immunity, as we have told, snake bites and all, anti-venom injection from horses or rabbits, right? But this also, because of the PETA and everything, now even we are not getting proper snake anti-venom, because earlier it was in Chennai, where horses were injected with minute doses of venom from the snake, and that particular anti-venom or antibodies were been obtained and injected. Now, immunity is very short-lived. Wherever you could see that antibodies are being injected, antibodies are being provided immediately. Whenever the antibodies are injected, that gives us protection, and as they give protection, but it is short-lived, maybe a month or two so. Then the host immune system does not respond to the antigen. Fluid that remains after blood has clotted and cells have been removed. Fluid that remains after blood has clotted and cells has been removed. What is serum? A blood, if it is taken, a blood is been taken, and if you centrifuge, or if there is a cut, what we could see that in the cut or wound, a yellow kind of formation happens, which helps us in protection. So what is that? That is called the serum, which has got antibody, and it helps us in protecting. Anti-serum, serum containing antibodies to a specific antigens. Serum containing antibodies to a specific antigens. Obtained from injecting an animal, horse, rabbit, goat, with antigen, snake venom, botulism, diphtheria, and toxins. So you could see, for all those, we require antibodies. We require antibodies. Serology, the study of reaction between antibodies and antigens, that is called serology. Then the gamma globulins, these are the very important gamma globulins, which is the most important part, and these are the one which is like $1 million project, or this is called $1 million globulins, antibodies, which helps us in protection. And this is how you could see, like Covishield, Covaxin, and everything, they had been maintained with. Then the serum sickness, disease caused by multiple infection of anti-serum. See, many a times, therefore, injection of anti-serum, many people, they can't withstand plasma therapy, because it can cause backfire, or it can cause multiple problems because multiple serum or no antibodies may be there. They may cross react and they can cause fever, kidney problem, joint, but in fact, they are rare today. So this dual of immune system, humoral or cellular immunity. What is humoral? As I have already explained, so let me not tell this much more. Then B cells already we have told, extracellular. Yeah, this is the one which we must learn is antibodies are produced by B lymphocytes, right? Antibodies are produced by B lymphocytes. So what it happens, you could see that there is a fetal liver that is our spleen or bone marrow stromal cells. From there we get lymphocyte stem cell. From there it goes to T cell. They mature, they are called T cell, and they go to lymphoid tissues and they become B cells. And this is the lymph nodes are the one or the blood where the war happens. Now coming to this beautiful structure, which is the key for us to understand antibodies. What is this key? Antibodies are proteins that recognize specific antigens. So here you should understand, this is just an antibody molecule structure. And this is the one which is called as a variable region. And this is the one in antibody called as a paratope. And this is the one in antigen which is called epitope. So this paratope and epitope interacts and then they get detached from here. So the moment paratope, this is a paratope, reacts with an epitope, the reaction goes through this and now the stem region, and then from the hinge region they get detached with. FAB and FAC happens. So usually what we tell structure of antibodies is Y-shaped. This is the how the structure looks, more detailed structure. And this was, I was been telling, right? Carbohydrate moiety is there. Glycoproteins, these are proteinaceous in nature. These are the polypeptides. And this is the carbohydrate in nature. So this is, you could see this. This is an antigen. So they should be specific. Biological molecule, they can't be altered. So they are specific to the sites. Then they have variable light, variable heavy. So this is the paratope and epitopic site. They will bind. Then the reaction comes. And once the reaction comes, they split. And then the molecules start interacting with. So they have the hypervariable region. They have complementary region. So later on, you will be studying this. So these variable regions, and then the cell-mediated immunity. The same thing, cell-mediated immunity also goes that way. The cell-mediated immunity is also like that. But in the difference between humoral and cell immunity is there is no antibody. Directly the cell tries to engulf or become cytotoxic T lymphocyte, and then they engulf the antigens. Now coming to the antigens, so much we have been telling what is antigen, what is antigen. So what they are the one which provokes, right? They are the one which provokes an immune response in specific lymphocyte as in foreign or as a not a normal constituent of a body. Don't take ever that your cell will trigger immune response. It will be always a foreign substance or it will be a cell which has become altered. They will try to trigger the response. Then most of them are protein in nature, and already we have told they are 10,000 or more kilodalton molecular weight. Then they will be microbes in nature like capsule, cell, wall, toxins or viral capsids or flagella. So you might have seen during typhoid, initially if a person is affected with typhoid, it will be somatic, but later on, if the person is getting affected with or it is a prolonged one, it will be the flagellar one. Then you have this non-microbes like pollen, egg white. Many people, they can't take brinjal eggs, right? Or they can't take animal proteins. And they have the surface molecules from the transplanted tissue. Therefore, the tissue rejection, many people you could have seen, they transplant, the graft has been taking place, but suddenly what they could see, the operation is successful, but the rejection happening because of the transplanted tissue has not been compatible. And this compatible is been recorded in form of MHC molecule, major histocompatibility. So here what we do mean hapten. So everyone we have come across that during our polio, right? Polio vaccination, the moment a drop of polio has been administered to our mouth, we start crying. Why? The reason is the attenuated or no live attenuated or heat killed virus, when it enters into our mouth, but because of attenuation or heat kill, they lose the property of antigenicity. So that molecules, what we do, those molecules are being added with certain called as adjuvants like alum. As we add, the molecular weight is enhanced up to 10,000 kilodalton, and then they can readily trigger or get the property of antigenicity or anti-reactivity. So a coupled with a carrier molecule, a carrier is required. Take an example of a pen. You have a refill, but you don't have the cover. So if you with a refill, you can't write. You may write little bit, but you can't write for quite longer time. So you require a body of the pen which helps aerodynamicity, which helps to hold, run fast. So that's why you require a carrier molecule. And the carrier molecule, as you could see, they can be polysaccharide, lipopolysaccharide, they can be. Then what is an epitope? As I have told, antigenic determinant site, small part of an antigen that interacts with an antibody, and then antibody will have a paratope. So this, the part epitope will bind to a paratope and the reaction happens. And each epitope is recognized by a different antibody. Different epitope will have a different antibody. So this is how you could see that. These are the microbial cells or viruses. These are the foreign bodies, pollens. So you could see that antigenic determinant sites, how they can be. So this is how all those happens. These are the different parts, antigenic determinant sites. This is how it happens. So the antibody can bind with this, the antibody can bind like this, the antibody can bind like this, or the antibody can bind like this. So they have, so like this, no, a virus particle, if you can see, you will find different kinds of antigens. Then there are autoantigens, molecules on cell tissues for which tolerance is inadequate. Autoantigens. Then you have alloantigens, one individual that are to another of the same species, or heterophile, or supra-antigen, potent T cell stimulator like toxic or allergen. So no, this let me antigen presenting cells. So always I can compare this with the our in body, like how a terrorist or a invader enters into a country or state, somebody catches and then brings to the takes them to the king's court. Same way in our body, when a foreign substance enters into our body, immediately there is an macrophage, neutrophil, dendritic cells. They act as an antigen presenting cells. The word itself indicates antigen presenting cell. That means how to present an antigen and what action has to be taken. It is been decided by the T cell, but to decide they have to be brought near to them T helper cell or TC cell by antigen presenting cell. This is how the reaction happens in the body. Once an antigen enters, as you could see, foreign, they will enter into the body, as you could see this process, right? They are been broken down, right? And then they are been broken down and then they are projected with MSC. Like this, it happens. Then helper T cells. So helper T cells, what they will do, they will get activated helper T cell, interleukin, and then they will form T cell and antigen presenting cell. This is, see, it looks very simple, T cell and antigen presenting cell binds. But here the complexity you can see, the moment MHC molecule binds with an antigen and then the T cell receptor recognizes there, CD4 tells them, yeah, the bond is very strong and it is correct bonding, go ahead with. Then the CD28 and B7 gives a signal for the reaction to happen. So this is how it happens, right? An antigen can have multiple different antibody can bind. Antibody A, antibody B, antibody C, and these are the different epitopic sites. So the B cell activations, right? All see, this is how the B cell activation happens. So this is how. So this image is the first one where you could see T cells get activated. And once T cell get activated, the B cell becomes what they do. B cell also process the same. They also bind with the same type of antigen. And then the B cell T cell interact with CD4, and then the activated B cells, you could see memory B cells and plasma cells. And these plasma cells are the one which secrete antibodies, and then these antibodies will kill the microbes. And these antibodies are from the plasma cells. Okay. So these two images are most important, and throughout, no, it happens and keep on happening in our body regularly. So what are these antibodies then? Now we have discussed about antigen. What are these antibodies? So these antibodies are called as, or there are of 5 types. IgG, they are called immunoglobulin gamma. The heavy chain, if it is gamma, we call immunoglobulin gamma. IgG, IgM, that is mu, immunoglobulin M, IgM, IgD, delta, IgE, epsilon, and Ig alpha, IgA. So each antibody had at least 2 identical sites. That means 2 paratopic sites. And these sites will have, like IgG will have 2 valence, like it can bind with 2 antigens. So they are monomer, as I have told. So let me show you the image here. Already this image has been shown. So directly I can tell you that they are a flexible Y-shaped. They are flexible like this Y-shaped. So this will bind with 1, this will bind with 1. And these are identical. So 2 Y-shaped larger, and then sidewise 2 smaller will be there, 2 smaller. So 2 light chains and 2 heavy chains. Identical they will be. And then this Y will have FAB and FAC, FAB and FAC. And then they have the constant region. This is the constant region. So when these two binds, the reaction goes like this through the stem cell. It goes into the cell, and then the cell start responding. That is FC region, monomer only, right? The FC region. So this is the one, FC region. So what you could see in the stem. Domain structure. So this is the one, let me tell you. So this is one heavy chain. So you could see that variable heavy, constant heavy 1, constant heavy 2, constant heavy 3. Right? And based on this heavy chain, they are being recognized as alpha, delta, epsilon, gamma, and mu. IgA, IgD, IgE, IgG, and IgM. And this is the light chain. Variable light, constant light. They are depending on kappa and lambda. They are being categorized into. So there will be 2 identical, right? This is the molecular structure. And then they are joined together by disulfide bonds. Disulfide bond. That's why you could see that whenever there is protein degradation, cysteine residues are being released, which are sulfur rich. Intra-disulfide bond and inter-disulfide bonds will be there. And then they have the pepsine cleavage and pepine cleavage. So blue one is the pepsine and orange one is the pepine cleavage. So if it is a pepsine cleavage, right? Pepsine is a cleavage. If that breaks down, then it will give rise to, right? 1 FAB into 2 and 1 into FC, and this will give rise to 2 FAB and 1 FC. So this is FC and this is FAB. Antigen binding fragments, and FC is the constant fragment. So this is how you will get it. Then these are the different modules to show how the hypervariable region changes and they modifies themselves. And these are the different classes of immunoglobulins, these class, right? And these 5 classes, mostly this IgG, the first one, IgM, IgE. These are the 3 major ones which plays a major role, with little bit of A, and D is little unknown. So what is immunoglobulin class? IgG. Immunoglobulin G They are the nearly 60 to 70% of immunoglobulin, and the structure is a monomeric structure. And as you could see, the percentage serum antibodies, they are around 80%. They are around 80%. Percentage serum antibodies, they are 80%. They are found in blood, they are found in lymph, they are found in intestine. Immunoglobulin G, and they are very specific. Then the half-life in serum is 23 days. Just nearly, no, you could tell that 7 days a week, 4 weeks. Oh, sorry, 3 weeks, around 3 weeks it comes. Then yes, complement fixation is there. Then the placental transfer. Therefore, you might have studied of erythroblastosis fetalis. If there is earlier days, now it is, anyway, we have anti-RH, everything are there. RH positive and RH negative. If RH negative marries a RH positive, the first child survives, but the second child dies because RH antigen enters the placental barrier and causes the death of the child in the womb itself. RH positive and negative. IgG formation, but now it's not, because already precautionary steps and anti-RH can be introduced prior on. Then known functions: enhances phagocytosis, neutralizes toxins, viruses, protects the fetus and newborn. IgG, therefore IgG is very important. Then IgM, this is also called as a millennial molecule. This is also called as a millennial molecule. They are pentamer in shape. So 5 times of IgG, and they are generalized, and they have only 5 to 10%. They are only 5 to 10% in the body. Location, either blood, lymph, B cell. Half-life is hardly 5 days a week. Complement fixation, yes, there is no placental transfer. Because of the higher molecular weight, there is no placental transfer. And the first antibody is produced during an infection, and they are the first one. Therefore, they are called as a generalized one. And what they do, they trap the antigens, agglutinating antigens. Then we have IgA, that is dimer, that is comes from the mother's milk, and they can be found in tears, saliva, intestine, blood, and serum. And there is no placental transfer, and they are localized mucosal surface. Therefore, mucosal transfer from mouth till the anal part, they helps in localized protection, digestive tract. Then the IgD, it is the less known, 3 days and hardly 0.2%. And IgE, it requires a extensive study, and moreover, Ig is the one where we can tell that hypersensitive 1, 2, 3, and 4 reactions, how allergic. Initially, you might have seen we play with the chalk and everything, nothing happens. But nowadays, little more you play with the chalk, you find that you start getting affected with chalk, dust allergy, cold allergy, now even cold allergy, right? Dust allergy. So all those allergy comes under this picture called IgE, allergic reactions, and even the lysis of the worms. So this, if you could see, if we take a total serum and then we can spread out into, we will find that IgA, Mg, and all total serum can be split up into alpha 1, alpha 2, beta, and gamma. Like this we can find out. So ultimately what we understand from here, that primary and secondary response is most important. A primary response makes us understand that is the first exposure and to an antigen. And the second one is the one which gives us immunoglobulin G, and we become more specific to that. So the primary and secondary response plays a major role. So therefore you could see here IgG, IgM, IgG. So always you could see that IgG will have in initial stage, you could see IgM comes first, total becomes like this, IgG comes little later. But in the second exposure, you could see that always IgG becomes more, IgM becomes less there, second exposure, but to the same antigen. And this is the world where we have to understand, if you go for blood grouping, if you go for any kind of vaccine production, it is called as a monoclonal. What does this monoclonal means? It means that the Y-shaped, this one I have shown you, right? Y-shaped, they binds with two different antigens. Now, no, they has to bind with the same type of antigen. That's why they are called as monoclonal antibodies. They are called as monoclonal antibodies, single clones. And this is the one by mice. They have been produced, and no, they can be proliferating. They are being coupled with, they proliferate, differentiate, same type of, and at any point of time they will produce the antibodies. So Vidyut sir, so can I leave for or I can speak some more time?
Sir, you have to take another 5 to 10 minutes, sir.
Okay, done.
So, if you don't mind, in between the 5 to 10 minutes,
Ah,
please send today's topic 2 questions, distributed 2 questions, sir.
okay, fine.
Ah.
See, this is one which I had been telling, right? Stimulating, this is overall. So you could see the antigen receptors, antigens binds with, and then this is the clone of plasma cells which produces the antibody, and this is the memory cell remains. So these memory cell and plasma cell we are juxtaposed between these two to give us the resistance. So other than this, nothing works. So ultimately it is you, your body, whatever medicine you take, whatever, they are all temporary. If we improve our immunity, then only we are safe. So if we don't improve our immunity, what will happen? Our cell starts behaving differently. Then we'll start becoming like programmed cell death, falling away. And then human body around makes 100 million lymphocytes every day. Wonderful, right? 100 million lymphocytes every day. If an equivalent number doesn't die, we'll develop leukemia. See, we are walking on the sword's edge. This side we fall, that side we fall. This side if we fall, we'll get hurt in the stone. This side if we fall, there is water. So this is how it is, and edge of the sword we are working with. So human body makes around 100 million lymphocytes every day, and if an equivalent number doesn't die, leukemia, leukopenia, and all those comes. So the B cell that do not encounter stimulating antigen will self-destruct and send signals to phagocytes to dispose of their remains. So therefore, the phagocytes, the number, so usually when there is an infection, you could see there lots of antibody, therefore inflammation happens and lots of WBCs comes into the picture, and if they are not disposed, therefore in COVID, why I am relating COVID, it is easy to correlate. People have started giving steroid, but when many doctors, they have given steroid in COVID, after a week, when the virus after a week life cycle is over, immediately our body cell, where number of cells were very much high, 10 times more, they has to be eliminated. But as their body has become low, they were not able to eliminate. Steroid was used to suppress the inflammation. Apoptosis, programmed cell death. So how are the receptors? So ultimately it is the receptor, and we tell, right? Our reflexes are good, our receptors are good. These receptors are the one where the cell matures, they pick up the cell surface receptors, and they are packed with phosphorylation, glycosylation, and it is inserted into the cell membrane. And these acts as an antenna molecules, like your antenna. See, you have seen your mobile. While calling suddenly in a remote area, what you do, you try to lift your phone above, or you try to bend towards the window, or you try to see that at least little bit mobile is outside the window and you speak. That means it catches the signal. Same way in your cell, the receptors can't pop out. But what you have to do, your cell must have a sufficient or good quality of receptor. As a result, what will happen, if receptor is there, they will pick up the signals. So lymphocyte use around how many genes? 500 genes to produce a tremendous variety of specific receptors. And each receptor has each function. See, during quiz competition, you might have seen many people, immediately, most of us know the answer, but promptly answering becomes difficult. But whenever we are outside the, no, if we are not competing, we are as an audience, promptly it comes. That means there is a receptor which correlates between the conscious mind and subconscious mind. So this receptor plays a major role in understanding and triggering the immune response. See, these are the different kinds of receptor, how it comes. Genes for the receptors, how they are being attached from the nucleus, nuclear membrane, and then they come out. Golgi complex carbohydrate receptor proteins are being attached, and they are being packed, and they are released. And this is a beautiful complex. I am covering a little briefly. Hope so next time when we get time, we can briefly elaborate MHC, because this MHC2 is present in chromosome number 6 and 10. And this is the one which determines that we can donate organ to somebody else, or cell or tissues. Therefore, you might have seen, especially in the bypass surgery, the vein from the leg is been taken and from the same person taken and put as a bypass, because they don't want to take a risk with somebody else, because heart operation is a critical bypass surgery. The operation may be successful, but the patient may die because of the allergic reaction or non-compatibility. Therefore, this part that is called as a major histocompatibility complex, or human leukocytic antigen HLA, plays an antigen which can trigger immune response in somebody else. And therefore, classical example is Michael Jackson. You could have seen he was under frequent skin surgery, because once he started with skin surgery, there was little bit problems happening with the non-compatibility, and to overcome that, he underwent many surgeries like that, which landed up in problem. And so therefore, you must understand that their MSC is 1, 2, and 3. So 1 and 2, this is chromosome located in chromosome number 6, clustered in a multi-gene complex of classes 1, and they are, right? Complement system is present in 3. Class 1 is for cell-mediated immunity. Class 2 is mediated for humoral immunity. So this is how, in chromosome number 6, how these classes of genes and MSC2, as you could see, MSC2 means both recognition of it will have alpha 1, alpha 2, beta 1, beta 2. They go deep inside the cell membrane, whereas in MSC1 molecule, that is CD8, you have only one going inside. The reaction responses will be little less compared to the 2. Then the function of MSC, that they require T lymphocyte in class 1, whereas in class 2, yes, they are here. I will end. Class 1 are the one where markers that display unique characteristics of self-molecule regulation, class 1. Class 2 is the one react with the foreign antigens, and they are looking into primarily, right, like macrophages, neutrophils, dendritic cells, and different, they are called like scavengers, or they are called as purifiers, or like bouncers. They eliminate the unwanted cells or non-cell cells from our body. And the class 3 are the one which helps in secreting the complement system, C2, C4, which in turn helps in C1, Q, R, and S binding. And then from there they go to C3, then 5, then 6, 7, 8, and later on C9, along with C6, 7, 8, and then C5b, and then they form the triggering or the creating a hole in the cell surface, and it helps in killing the microorganism. So in fact, the health and human health and disease is a big part. I just touched a brief part of human health, that these are the cells which regulate our health. These are the antigens, antibody, MSC. They are the one which always we have to enhance and which we have to take care of, so that they can protect us from the diseases. And this is very, very important. Either it can be HIV, either it can be influenza, it can be virus, it can be bacteria, it can be pathogen, it can be worm, anything else. You take an example, everywhere these cells are involved. So now I request Vidyut sir to check out if there is any question, or we can have a few more interactions if time is there.
Dear students, please asking any questions from the today's topic. ಯಾವುದೇ ಕ್ವಶನ್ಸ್ ಇದ್ರು ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಮಾಲಿಕೆ ಮೇಲೆ ವಿದ್ಯಾರ್ಥಿಗಳೆಲ್ಲರೂ ಕೇಳಬಹುದು. ಸೊ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಯಾವುದೇ ಕ್ವಶನ್ಸ್ ಕಳಿಸಿಲ್ಲ ಸರ್ ವಿದ್ಯಾರ್ಥಿಗಳು. ಸೊ ಲೇಟರ್ ಆನ್ ಐ ಸೆಂಡ್ ಥ್ರೂ mail, sir.
No problem, sir. So I would like to, from this, I would like to pose 2 questions or 3 questions to the student. First of all, I like to understand not only from their academic point of view, but from in general point of view, from their health, what is innate immunity and what is acquired immunity? Because if they understand innate immunity, they will come to understand what their body is. And acquired immunity, to understand, they need to check their vaccination chart. That is one. The next question I'd like to ask them, what are the properties of antibodies? Or what is antigen and what is antigens and its significance? Because the antigens has got certain characteristics. So briefly tell me out, what are antigens?
Questions.
Yes, sir.
Please tell me, sir.
Yeah. So antigens and its significance, or antigens and its characteristics. So always we have a question. Many a times during COVID-19 we have heard of, sir, this has got more efficiency, this has got less, this has got 94%, this has got 95%. So therefore we must understand what is efficiency. That means the antigen, the properties of antigen plays a major role to design a vaccine, because that is the one which goes into our body and triggers the immune response. So antigen and its properties. So what are antibodies? Characteristics of antibodies, antigen and its significance, and innate and acquired immunity. I think if you learn this, you can learn any war within and outside your body. So these are the more questions. So I would like to pose to the students so that they can know, not only for their chapter, this health and human health and disease, even for their future, it will be helpful for them.
So end of this session, please, if you don't mind, please send me through WhatsApp, sir, these 3 questions.
Sure, sir. Sure, I'll send you that.
Again, I send through the students by group and chat box, sir.
Sure, sir.
So thank you, sir. ತಾವು ಕೊಟ್ಟಂತ ಇವತ್ತಿನ ಹ್ಯೂಮನ್ ಹೆಲ್ತ್ ಅಂಡ್ ಡಿಸೀಸ್ ಬಗ್ಗೆ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಬರೀ ಸರಳವಾಗಿ ಹಲವಾರು ವಿಚಾರಗಳ ಬಗ್ಗೆ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ರಿ ಸರ್. So it's an immense pleasure to thank you, Santana sir, for this wonderful speech on human health and disease. This talk has given many information to the students. I gratefully thank you, sir, from KSTA and personally, sir. And I also thank our KSTA CEO, sir, Dr. Ramesh, sir, for this encouragement to organize this talent search program. I am gratefully thank you, sir. And I also thank our chairman, Professor Ayappan, sir, and colleagues, technicians, staff members for their participation to conduct this program. Once again, thank each and everyone for their participation. Okay, thank you, sir.
I thank you wholeheartedly for giving me the opportunity. Hope something has helped the student to understand this part. Thank you, sir. God bless you, sir. Thank you. Stay safe.
Thank you very much, sir.
Thank you. Thank you, sir.
So, ನಾಳಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ ಮಹೇಶ್ ಬಾಬು ರವರು ವೇವ್ಸ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನಡೆಸಿಕೊಡಲಿದ್ದು, ಎಂದಿನಂತೆ ವಿದ್ಯಾರ್ಥಿಗಳು ಪ್ರತಿಯೊಬ್ಬರು ಹಾಜರಾಗಬೇಕೆಂದು ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಇಲಾಖಾ ವತಿಯಿಂದ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ಮತ್ತೊಮ್ಮೆ ತಿಳಿಸುತ್ತಿದ್ದೇವೆ. So, Munandar, I thank everybody. Thank you, sir.
Thank you, sir.